Welcome to the study of histology or microscopic anatomy. We're going to begin with a basic introduction to cell and tissue structure. These are the learning objectives that you should use when studying this material. So let's start by taking a look at eukaryotic cells. Why should we really care about cells and cell structure? The reason is all normal physiological functions and pathological alterations of these functions are the result of activities of cells that form the tissues and organs of the body. The basic premise of histology or anatomy in general is that form follows function and malformation follows malfunction. Therefore, it behooves us to really understand how the structure and functions are connected. The changes in the organization and the function of the cells as a result of disease processes, be they congenital or metabolic changes, or even infections or injuries, are the basis for all the illness and disease that you're going to be looking at as physicians. So cells are the simplest organizational structure that has all of the attributes of life. Consequently, if you're alive, it means you're either a single cell or a community of cells. So what is a healthy, functioning community of cells? So I'm showing you some images here, which you won't be familiar with yet, most likely. At the top, you'll see a normal mammary gland. The cells are gathered into groups. You can see at the higher magnification that they form these little tubular kind of circular structures. And that's where they're producing their product. This is a gland. It makes milk. So they have to have a particular structural relationship to one another and to the underlying tissue which is supporting them in order to function properly. When you have changes in the metabolism, such as in uncontrolled cell growth, tumor, metastasis, whatever, you have a change, consequently, in the way these cells are organized, and they're no longer functioning properly. You can see in the high mag on the right that the lumen is completely filled with a lot of debris. The cells are no longer lining those nice little tubules, so they can't really produce their milk properly. And this uncontrolled growth, of course, can lead to tumors that can be life-threatening. So cell organization and the function are interlinked here, and when you compromise one, you compromise the other. So this is an example of a pathological community of cells. So how do cells function normally? They utilize a number of subcellular structures called organelles, each of which has specializations that carry out specific functions. What are the cell organelles? They are intercellular structures which may or may not be membrane bound, right? And you can identify them in electron micrographs. And we're going to do that in this first unit, although not within this series of videos. You'll do that within a series of electron micrographs that will be provided to you. Most organelles have that membrane structure, although some do not. And we'll take a look at all of those variations as we go through. How do cell organelles contribute to the cellular functions? Well, there's a number of functions that cells have to perform in order to live, in order to grow, divide, survive, maintain themselves, and so on. And those are listed here. And there's a number of organelles that are involved in these functions. However, it's not really a one-to-one relationship. In other words, all adherence is not carried out by one particular thing, or all secretion is not carried out by one particular organelle. Instead, it's a more complex relationship. You have a number of functions that rely on several different organelles in order to be carried out efficiently, and you have a number of organelles that participate in several different processes. So in this series of videos, we'll go through a review of organelle structure and function. However, you'll also be provided with an organelle self-study, and you will be responsible for the material within that self-study. Additionally, as I mentioned previously, you will be provided with a number of electron micrographs which are annotated, and you will be expected to identify cell organelles on those electron micrographs in this block. So, starting with the very outside of a cell, the first organelle that is encountered is the plasma membrane. The plasma membrane functions to separate the cells, or what's inside the cell, from what's outside the cell, so it compartmentalizes that cell. It has specific receptors or proteins on its surface, and it can transduce signals from the outside in and from the inside out. It controls all of the movements of materials into and out of the cell, sometimes passively, sometimes actively, and that will be studied in much more detail by the physiologists. It controls adhesion. In other words, some cells adhere to one another and some cells adhere to their matrix, which is the surrounding material. And that adhesion process is very important to the survival and indeed to forming communities of cells in general, so that they can function together. So let's take a closer look at the plasma membrane and how it's put together. On the left, you can see a model, and this is familiar to many of you who've had basic biology or even cell biology. This is called the fluid mosaic model of membrane structure. And in this model, you can see that the membrane is comprised of a bilayer of lipid molecules called phospholipids, and these have a hydrophilic polar head and a hydrophobic fatty acid chains or tail portion, and that is directed towards the center of the bilayer. The membrane is decorated, so to speak, with a number of proteins and carbohydrates on both the inner and outer surface, and these proteins can be either peripheral, attached to the polar head portions of the lipids, or they can be embedded within the bilayer, and those are integral membrane proteins. And those are the proteins that would act as transducers, as transporters to get things across, as linkers to various other structures within and without the cells. So really those proteins are very important to the function of the plasma membrane. So on the right here you can see a really nice micrograph of the lipid bilayers with the separate hydrophobic interior, which is poorly staining, and the head regions, which are darkly staining. So you can see both of the lipid bilayers there.